Hello everyone, and welcome to today's webinar, Critical Reagent Characterization and Candidate Selection powered by the UNCLE platform. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Unchained Labs. To learn more, visit unchainedlabs.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Dr. Ron Bowsher, Partner and CSO at B2S Life Sciences, Adam Kinne, Scientist, Custom Reagents for B2S Life Sciences, and Dr. Kevin Lance, Marketing Manager at Unchained Labs. Kevin, you may now begin your presentation.
Thanks for that introduction. I'll be introducing and giving some more background to the thermal unfolding and aggregation parts of this webinar and the instrument used for much of the analysis that you'll see. So when dealing with a hard choice between a lot of options for proteins as critical reagents, picking out just one best option can leave you feeling a bit like the statue on the left, overwhelmed. In this webinar, myself, Ron, and Adam will talk about how characterization using the UNCLE from Unchained Labs can help you ID the best performing options. When thinking about the stability of a protein, we can summarize it as two different types of stabilities, conformational and colloidal. Conformational stability, shown in blue on the left, is the ability of the protein to stay folded in its native state. Thankfully, examining protein fluorescence is one way to get insight into the conformational stability of a protein and is an easy way to examine thermally induced unfolding. Colloidal stability, shown in green on the right, can be thought of as the aggregation behavior of a protein. Gathering size information from static and dynamic light scattering can inform the understanding of a protein's colloidal stability. And both of these behaviors combine to paint a fuller picture of a protein's stability that you can use to pick the best option. So that leads us to the UNCLE and its ability to gather conformational and colloidal information to paint a fuller picture of protein stability. UNCLE is an all-in-one multimodal biologic stability platform that combines 3 detection methods in 12 applications to help you understand the stability and aggregation behavior of your proteins and critical reagents. To test any of these applications, only 9 microliters of sample is needed, and up to 48 samples can be run at once. Those 3 detection methods on the UNCLE are full spectrum fluorescence, static light scattering, or SLS, and dynamic light scattering, or DLS. UNCLE can see the full spectrum for fluorescence to give you the option to look at protein intrinsic fluorescence, which evaluates usually tyrosine and tryptophan fluorescence, or use any of an arsenal of fluorescent dyes. Simultaneously with that fluorescence measurement, you can use static light scattering to monitor how the same sample aggregates. UNCLE uses SLS at 2 wavelengths so you can track small and large particles at the same time. Dynamic light scattering, or DLS, is UNCLE's 3rd detection method and is a highly sensitive technique to give you details about your protein's size and size distribution. So besides gathering size data, DLS is powerful for understanding what's in your sample, and it can be used to quality check proteins either independently or before you start a thermal stability experiment. Now I'm going to introduce one of the key features of the UNCLE, which is the Uni Sample Folder. The Uni is composed of an array of 16 quartz microcuvettes that are easily loaded with a pipette. The high-grade quartz gets us great optical sensitivity for fluorescence and light scattering data, and the frame is an anodized aluminum that provides efficient heat transfer. Once you load each of the wells, the Uni is then clipped together in the blue frame you see at the bottom of the slide, where the silicone seals provide a nice tight seal around each of the Uni cuvettes. This means that you can read a Uni right away, or you can pop it into and out of an incubator or fridge and take time points over a long period of time. Now, a little bit of detail on the underlying phenomenon of protein fluorescence. Here we're looking at the 3 fluorescent amino acids in a protein, tryptophan, tyrosine, and phenylalanine. In blue, we have the absorbance spectra of each amino acid. So when we hit these proteins with a 266 nanometer laser that's in the uncle, it will absorb light from that UV laser and fluoresce it, which is what's shown in green. Altogether, all of those amino acids inside of a protein combine to be the intrinsic protein fluorescence. And the important part of this is as proteins unfold, that intrinsic protein fluorescence will change. So you'll actually see changes in the combined green fluorescence behavior. We see that change here on the slide. So over a heat ramp, Uncle will excite and read the full spectrum fluorescence of a protein sample at numerous temperatures. Each line in this graph is a fluorescence read of a protein excited with that 266 nanometer laser, starting with the read at 50 degrees Celsius and going all the way up to 95 degrees Celsius. You can see by following the curves from top to bottom how they shift to the right side of the graph, which are the longer, redder wavelengths. This phenomenon is called a redshift and occurs because the protein is unfolding. Buried tryptophan residues have a shifting fluorescence emission as they move from being buried in hydrophobic pockets to being exposed to an aqueous environment as the protein unfolds. This protein shows a classic redshift pattern, but you never know how each protein will behave until you've tried it. Some proteins shift to the left, called a blue shift, while others can decrease in fluorescence without a shift. With Uncle's way of collecting full spectrum fluorescence, you have all of the data and you don't have to worry about unexpected protein behavior. And for proteins without tryptophan and tyrosine residues, Uncle also has a 473 nanometer blue laser that can be used with cypro orange dye to excite fluorescence and determine unfolding. The analysis method we recommend reports the barycentric mean of that full spectrum fluorescence. This is shown on the left. It's the wavelength that evenly divides the area under the curve as a kind of spectral center of mass. We found that this analysis method is the least susceptible to noise since it integrates a large range of the fluorescence spectrum. Analysis of the protein's fluorescence then is used to show unfolding and identifies inflection points as melting temperatures of that protein. We see results of that kind of experiment here, where in blue we're looking at the BCM data, and in green we're looking at static light scattering data. These pieces of data are gathered simultaneously so you understand protein thermal stability and aggregation information in the same experiment on the same sample read all at the same time. So fluorescence in this case, the blue data, shows our proteins unfolding, and we can see inflection points that are identified as melting temperatures at TM1 and TM2. The SLS data in green shows the aggregation behavior of your protein and reports the temperature when aggregation begins as a TAG. You notice how the TAG event lines up with TM2, which is a link between the second unfolding event and the aggregation event of the protein. So together, the two protein tools of intrinsic fluorescence and SLS give you a good understanding of how unfolding and aggregation is occurring in response to thermal stress of protein. Dynamic light scattering, or DLS, which is Uncle's third detection method, is a highly sensitive technique to give you data on your protein size and size distribution. Here we're looking at some example data of a monomeric sample in blue, a sample with some aggregation in green, and a sample with significant aggregation in yellow. I want to point out that the x-axis is in a log scale. The metrics that are reported from DLS data are typically the z-average diameter, which is a weighted average of diameter for the whole sample population, and a polydispersity index value, or PDI, which is the measure of the width of the size distribution. Higher PDI values tell you polydispersed samples of many different sizes, and lower PDI values tell you of a monodispersed sample of a single size. Looking left to right across the slide at these samples, we see an increasing trend in the z-average diameter, which agrees with what we know about the sample state as aggregation increases. Likewise, the size distribution for these samples widen as you look left to right. So since higher PDI values indicate wider size distributions, PDI would also show an increasing trend here. In this case, the sample on the left is actually done at close to room temperature, and the temperature increases to the mid 70 degrees Celsius once you've gotten all the way to the right, which is how we drove aggregation for this sample. So using these tools can get you from a multitude of choices to finding the option that you really want. And now, Ron and Adam will introduce B2S Life Sciences and speak about Uncle's role in critical reagent characterization.
Good afternoon, everyone. Ron Bowsher here. It's my pleasure to talk with you today and my colleague Adam Kinne about the the application of the Unchained Labs Uncle instrument as an advanced protein characterization tool in our toolbox for evaluation of critical reagents. My background in this business is spanned roughly 30 years in pharmaceutical industry and about 20 years after retirement. And the focus of my career has been mostly around biotherapeutics. And today we have a business, B2S Life Sciences, that I'm going to offer a few comments about. Okay. To begin, our focus is on critical reagents, and I like to talk about the criticality of critical reagents. And what I mean there is this: the definition is the state or quality or the degree of something being of highest importance. The FDA today defines critical reagents as a requisite component in an assay, and as with the emergence of biotechnology-derived drugs, there's greater reliance today on critical reagents, and commensurate with that greater reliance is a need for good characterized and well-behaved critical reagents, hence the mission for B2S Life Sciences. To kind of reinforce the importance of critical reagents, there was several years ago, there was a flurry of activity around the reproducibility crisis, which brought greater awareness to the concern around the reproducibility and of the quality for commercially derived antibodies and critical reagents. Hence, a need for the consumer to provide better characterization to ensure that they're suitable for their intended purpose. In addition, over time, the FDA and the EMEA have both increased their awareness, their requirements, expectations around how we manage our critical reagents for supporting ligand binding assays. And as shown in this slide, there's verbiage around how those requirements have evolved over time. And the latest FDA guidance document, again, refers to critical reagents as requisite components that require management as well as good documentation. And as part of that requirement, we offer services to help characterize and manage critical reagents to ensure that they meet regulatory expectations. Our mission here at B2S Life Sciences is the design, the generation, the purification, the characterization, and life cycle management of custom critical reagents, and with complementary downstream activities in assay development. But the goal here, ultimately, for critical reagents is to enable successful regulatory submissions of biotherapeutics. So we're providing custom services to our clients for their critical reagents. And in order to do this, we rely heavily on the Uncle instrument for advanced characterization. The focus at B2S is commensurate with the emphasis on critical reagents. Our emphasis is upstream from typical bioanalytical CROs. So we're focused on reagents and assay development as opposed to being focused or emphasizing sample analysis. So we have a tiered approach that we have adapted at B2S. And the tier one, our standard characterization for a critical reagent includes an assessment of concentration, assessment of purity, an assessment of an incorporation ratio or conjugate testing, if it's a conjugate, and then complementary with that is an activity or affinity assessment for binding or what have you. When we need advanced characterization, when issues arise, when questions develop, we then have a set of advanced tools in which we can assess size heterogeneity, stability, purity, and binding kinetics. And as I said, the UNCLE platform is a key element in that advanced testing. What I want to do now is turn it over to Adam Kinne, who's going to offer a couple of very good case studies where the UNCLE instrument has proved to be highly valuable in being able to make time-efficient, data-driven decisions about our customers' project.
Thank you for that introduction, Ron. Keeping with the theme, I'm going to run through my background very quickly. I'm a scientist at B2S Life Sciences in the Custom Reagents group. Prior to coming to B2S, I had 8 years of experience as a synthetic chemist engaged in small molecule work, both organic and inorganic species. And then my time in the life sciences industry is a little bit newer, focusing on protein chemistry and critical reagents. So it wasn't a huge leap from small molecule, but I've since moved a little bit upstream into purification and characterization. So today we're presenting 2 case studies where the UNCLE instrument has aided our characterization through advanced characterization methods and what those outcomes were and key learning points from each case study. So the first case study examines the advanced characterization of one enzyme, which was produced and provided to us by 2 vendors. We had reason to believe that the 2 isozymes differed in biochemical behavior and warranted a closer look. So we chose our advanced characterization methods accordingly and gathered data to make our conclusions. So on this slide, I present the study design for this case study. We were seeking to learn 3 key things about the enzymes: what composition, stability, and activity were. So on the composition point, we were looking to tell what was in the samples that we received. The stability, we wanted to examine it based on how well the enzymes would respond to stresses. And the activity, we wanted to measure how well enzymes bound to the antibodies which we're using in the biochemical assays. So for the composition and stability, we used the UNCLE instrument, and for the activity, we used the Octet instrument, which is a BLI instrument produced by a different manufacturer. One of the So moving into the data analysis portion of this case study, we first analyzed the vendor 1 enzyme using the size and polydispersity method on the UNCLE platform. This is a dynamic light scattering method, so DLS, and this generates 3 plots in our configuration. So on the left, you have the intensity distribution, which gives you the hydrodynamic diameter that Kevin introduced earlier, and the amplitude on the y-axis. On the right-hand side, top, you have the correlation function, which is a mathematical representation of the scattering pattern within the sample. And then on right-hand side bottom, you have the mass distribution, which is a corrected representation of the population within the sample. So moving on to the vendor 2 data set, you can see that the hydrodynamic radius or hydrodynamic diameter is shifted much to the right, indicating that there are a large portion of rather large particles in this material. What that means to us is that this material was probably aggregated as received. So if you look at the mass distribution, it's all over the place and uninterpretable. But the correlation function and the intensity distribution indicate that by and large, the predominant species are large particles. So one criterion which Kevin introduced but was not evident on those previous slides is the PDI. So that, again, that's the polydispersity index, and it is a measure of the uniformity of a sample. So if you look at the vendor 1 material, it has a lower PDI, which indicates higher uniformity. And if you also look at the mode diameter of the first peak, that would be corresponding to the monomeric protein, is much smaller, so under 10 nanometers for the diameter. And the mass percentage of it is nearly 100%. But with the vendor 2 material, what would be referred to as the peak 1 in the vendor 1 material is absent, and the peak 1 in the vendor 2 material has a diameter of greater than 1,000 nanometers. And there's also a greater polydispersity index indicating poorer uniformity. So after we collect the DLS data, we often use the same samples within the same uni to perform a TM study. So this is thermal stability by way of melting point or melting temperature, which we often shorten as TM. And as Kevin mentioned in the top of the presentation, this is essentially measuring the intrinsic fluorescence of a protein as a function of temperature. So examining the data for the two vendor enzymes, again, we see a stark contrast between the two data sets. On the left-hand side, we have the vendor 1 material TM curve, which shows the normal sigmoidal morphology. And then on the right-hand side, we have the vendor 2 TM curve, which essentially looks like noise. So we don't get a real TM value for that. The melting point for the vendor 1 material was about 58 degrees C, and essentially what we concluded from examining the vendor 2 material was that it was likely unfolded or aggregated as received, which is consistent with the DLS data. And the final point that we wanted to look at in this case study was the antibody binding selectivity. So for this one, we used the Forte Bio BLI instrument. Essentially, this instrument measures the changes in interference patterns on a surface as a function of ligand binding. In this experiment, we immobilized the enzymes from vendor 1 and vendor 2 onto a biosensor, and then supplied monoclonal antibodies against the enzyme to the biosensors and measured the interference patterns. So if we break down these plots into left and right-hand side, the left-hand side is measuring the association. So you have a growing signal as more antibody binds to the enzymes. And then on the right-hand side, you have dissociation, where signal decays as the antibody starts to dissociate from the biosensor. And then if you compare the blue and red traces, blue traces being the vendor 1 material and red traces being the vendor 2 material, you notice that the association values were much higher for the vendor 1 material, and the dissociation curves are also correspondingly higher. So what that tells us is that more antibody was binding to the vendor 1 material and then dissociating away from it. And there's approximately a 9-fold increase in selectivity for the vendor 1 material. So wrapping up the first case study, it was an opportunity for us to use the UNCLE instrument to apply advanced characterization to 2 enzymes which were exhibiting differential behavior in biochemical assays. And the UNCLE instrument was the only tool that we had available here which could give us a clear answer on why this was happening. And by using the DLS and TM applications, we were able to choose the best isozyme for use in our critical assays. So moving on to our second case study, the goal of this study was to characterize and select or support candidate selection for a series of pegylated protein conjugates. And essentially, the idea was that we were supplied with a series of pegylated enzymes which advanced characterization was required. Again, we have a study design slide for this case study to define the scope of it. We received one unpegylated, or the parent unpegylated enzyme, and then 4 pegylated variants of it. They are referred to as candidates 1 through 4 for the pegylated enzymes, and candidates 1 through 3 have similar short chain peg modifications, where candidate 4 has a much larger branched peg chain. So that distinction will be important throughout this case study. And as with the previous case study, we used the DLS and TM applications of the UNCLE. So the first step we approached in this case study was to characterize the unpegylated enzyme first. And this sample and all of the candidates were run in parallel, thanks to the Uni Sample Folder that Kevin introduced. We were able to run all of them simultaneously. So if we look at the unpegylated protein data here, we have an average diameter of about 6 nanometers and a mode diameter of about 4.5, which is consistent with a monomeric protein. And then by looking at the mass distribution, we were able to determine that there was less than 1% high molecular weight species, and the PDI of 0.19 indicates that the enzyme was monodisperse, so highly uniform. So moving on to the numbered candidates, candidates 1 through 3, again, these are the short chain PEG modified enzymes. When we were examining the DLS data, they were all pretty similar. The diameters were between 13.6 and 14 nanometers for the average diameter, and then right around 13 for all of the mode diameters. The PDIs were all indicating monodispersed samples. So in this data set, they all looked more or less the same. And then moving on to candidate 4, you can see that the intensity distribution shows essentially no high molecular weight species. The mass distribution peaks all overlay really well, and the correlation function overlays pretty much perfectly. And if you, when you look at the diameters as well, they're significantly larger than the small PEG derivatives and larger still than the unpegylated protein, which is all consistent with expectation. And the polydispersity index of this one was also the best, indicating the highest uniformity of any of the numbered candidates. So bringing it all together, this is a table of the DLS data taken before the melting point determinations. You can see that we went from 6 to 13 to almost 16 in terms of the average diameters, and then the mode diameters go from 4.6 to about 13 to about 17. And this is so far all consistent with our expectations. So digging a little bit deeper into how the TM experiment works, essentially, once you have your plot of the BCM fluorescence, if you also plot the differential, so the solid red trace in all of these plots, whenever these hit a peak maximum, that defines a phase change or melting point in this experiment. One of the interesting things that came out of these data sets was that only the unpegylated enzyme showed a second TM, which from a structural standpoint, we're not able to say exactly what's happening there, but it appears as though the pegylation eliminates the second melting temperature. So if we look at the statistics for all these experiments, they all had less than 1% CV, which indicated good precision and minimal well-to-well variability. So if we move on to the individual plots of the thermal melting of the 4 numbered candidates, candidates 1 through 3 all look essentially the same. The TM is about 48, so 48.2 to 48.6. The candidate 4, again with the larger PEG, is slightly higher, so indicating a slightly improved conformational stability, but it also has another spectral feature where there's an additional equilibration event at the low temperature region, so about 15 to 45 degrees. There's a small hump in the BCM fluorescence. And then tabulating the data, we're able to compare all the metrics directly. The unpegylated protein had a slightly higher TM1 than any of the numbered candidates. This isn't anything really to worry about. This is just essentially to show that the TM1 is actually a pretty tight distribution here. So one other metric that we studied in this case, but didn't discuss in the previous one, is the static light scattering, or SLS, using the 266 nanometer laser. The purpose of this analysis is to detect and quantify small molecular weight aggregates. So that would be particles that would scatter 266 nanometer light. So these traces show some notable morphological differences. So in this slide, I've only presented candidate 2 and the unpegylated enzyme, but you can see that the aggregation temperature is shifted about 4 degrees to the right, indicating it improves colloidal stability of the numbered candidate against the pegylated enzyme. And this is probably the most important piece of data in this case, as this is probably the largest difference between unpegylated enzyme and any of the candidates. So if we look at all 4 of the numbered candidates, you can see that candidates 1 and 2 are about the same, and candidates 3 and 4 have a slightly improved colloidal stability, with candidate 4 being the best. And notably, candidate 4, again, is the large peg. So we were concluding that the larger peg modification led to the best improvement in colloidal stability. So moving on to the data summary, you can see that going from the unpegylated enzyme at 39, addition of any of the short pegs gave you about a 4 to 5 degree increase in stability, and then conjugating the enzyme with the large peg modification gave about slightly more than 7 degrees in improvement. And considering that the native protein melts at about 40 degrees, this is a significant improvement. So bringing it all together, to summarize the second case study, the different peg groups influenced both the conformational and colloidal stability. In this case study, the largest peg group gave the highest conformational stability as well as colloidal stability. And as expected, pegylation will increase the average particle size of any protein, and the pegylated derivatives also showed smaller PDIs, indicating that their particle size distributions were more homogeneous. And in terms of putting it into context, the UNCLE platform played a very crucial role in an early phase drug development project. So that leads us into our overall conclusions, which as you've seen over the course of the last two case studies, the UNCLE instrument has on many occasions proved to be a key instrument in our critical reagent characterization toolbox for our advanced characterization activities. And this especially comes into play when such investigations are needed to augment or sometimes even replace more traditional characterization methods. And the instrument has proved to be indispensable in our biophysical and stability assessments, especially whenever there are difficult to answer questions about client critical reagents. And our use of the instrument has been growing commensurate with the increasing structural diversity and complexity of contemporary biotherapeutics. So thank you all for your time in attending our webinar. We'll now field questions from the attendees, and presented here are all of our contact information if you would like to reach out and discuss any points beyond the Q&A session. Thank you for your time.
Thank you, Dr. Bowsher, Adam, and Dr. Lance for your informative presentation. We would like to now welcome Dr. Sanifar Abdeen to join us for the Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's get started. Our first question. During the TM measurements of the unpegylated enzymes, how many replicants did you use to calculate the percent CV?
So we typically use 4 measurements, more the better, but it's depending on the sample quantity. So in this case, we used 4 samples.
Okay, thank you, Sanifar. Looks like we've got a question here for Dr. Lance. How can we measure the TM of proteins / large peptides that lack aromatic amino acids?
Yeah, so as I mentioned kind of in the introduction section, we use a UV laser to excite aromatic amino acids, but not every protein has that. So it's pretty easy to use dyes like Cypro Orange and add those to a thermal stability experiment. And when you do that, you use UNCLE's blue laser, which excites that dye. And then as proteins unfold, that dye binds and fluorescence from that dye changes. So it's pretty easy to still look at those other proteins if they do not have aromatic amino acids.
Okay, thank you, Dr. Lance. We have another question here. What would be the minimum shift in tag or TM temperature between samples that you would consider significant to have a definitive conclusion, improved stability of decreased stability?
Okay, so I can probably take a first stab at this question, and then maybe a B2S can weigh in on their approach as well. So this is first kind of a statistical question, I would say. It depends on what's the variability in your sample. So kind of understanding if 2 populations are different. But if you just need a rule of thumb, then thinking about differences in 1 to 2 degrees Celsius that's where they start to become I would say put into different buckets between your best performers and maybe your next tier and your next tier.
Yeah, to jump on that a little bit, I would treat 1 to 2 degrees as where we start to consider things to be different. We usually report the values to 1/10 of 1 degree. So like Kevin was saying, it also kind of depends on the precision of the measurement.
Okay, fantastic. Thank you. Looks like I have another question here for Dr. Lance. you said that your laser for intrinsic fluorescence is at 266 nanometers. Isn't it tryptophan excitation usually at 280 nanometers?
So this is a pretty common question about the uncle. So tryptophan excitation peaks at 280 nanometers, but it's actually quite broad. So if you excite with the UV laser at 266 nanometers, what you're also getting is the ability to see tyrosine fluorescence as well, because that 266 will excite tryptophan and the tyrosine fluorescence, but by exciting at a lower wavelength, you're actually creating a little bit of space where that tyrosine fluorescence can appear at about 280 nanometers itself on the shoulder of a tryptophan excitation. If you excite with 280 nanometers, you're not going to see that tyrosine peak because it's going to be overshadowed by the larger tryptophan peak. So it's kind of a neat little way that we have to take a look at both tryptophan and tyrosine residues independently, while still exciting both with a pretty efficient excitation wavelength.
Thank you for that answer, Dr. Lance. We've got another question here. Let's see. On the evaluation of size and psi disparity, was the data generated during the heat ramp or at a different temperature?
So the data was generated at the room temperature. In addition to that, we also typically collect the DLS data at the initial and the endpoint of the TM or TAG analysis. So that is the 15 and 95, that is the typical. But the data which we presented was we measured the stability at room temperature.
Fantastic. Thank you, Sanafar. Another question for Dr. Lance. If proteins have the same melting temperatures, are you aware of examples aggregation temperature differs? Are you aware of examples aggregation temperature differs?
Yeah, definitely. So this actually kind of gets into the neat reason why having fluorescence and SLS on the same instrument kind of adds insight, is because you can have proteins where they have the same melting temperature, but the aggregation temperature can differ because maybe that aggregation event is tied to the first TM event in one protein, but the second TM event in a different protein. So it's definitely something that can happen, and it's important to measure both of those at the same time so you can have both options to rank your proteins by.
Great, thank you so much for that answer. Next question, what is the protein amount in milligrams needed for these kinds of applications?
So it depends a little bit on the size of your protein, but say, for example, if we're looking at an antibody protein, then we can go down to as little as 0.05 milligram per mil using only 9 microliters of sample. So we're talking about, micrograms of sample per I guess, well, per uni cuvette on the uncle.
Okay, great. I've got another uncle question for you. Have you used uncle for formulation development?
Oh.
So actually, I'll answer this shortly, and then I'll take it over to B2S. So basically, yes, uncle is used all the time by a lot of customers for formulation development, and I think, Rod definitely wants to speak about the B2S's experience.
Yeah. So what I would say is I think the uncle instrument is an ideal platform for doing that very thing, and is part of our kind of vision for helping our clients. We think that we can use the uncle to develop formulations to help stabilize other critical reagents. Unfortunately, we're so busy using it, we haven't had time to really do the formal DOE assessment, but we're confident that that's actually a major benefit of the technology.
Thank you, Ron, for that. Thank you, Dr. Bowsher. It looks like I have another question for the B2S folks here. Of the different capabilities that the uncle instrument possesses, which one has proved to be most beneficial for B2S custom reagent applications?
So based on our experience, we found that the thermal or the conformation stability and the aggregation stability data are very important. And in addition to that, these data can be easily correlated to their function of the critical reagent in the assay development standpoint in case of any pharmacokinetics or toxicokinetics for ADA assay development purposes. So for example, if your protein or ADA anti-drug antibody positive control is unfolded, then we would be directly see some sensitivity issue in your assay. In addition to that, if you see any aggregation, then it has been increased. You could clearly see in assay performance and high background. So I feel like those are the two most important factors that we routinely use for the UNCLE platform.
Thank you so much, Sanifar, for that answer. I have another question for B2S. What are the custom reagent capabilities of B2S?
Our range of services span antibody generation, design, purification, life cycle management of critical reagents, and we have a lot of tools in addition to the UNCLE that we use, daily for characterization. So I would say in a nutshell, it's the design, the generation, characterization, and then we have a very elaborate life cycle management proprietary system for providing life cycle management. So critical reagents, as Adam said, kind of is at the core of what we do. Thank you.
Thank you, Dr. Bowsher. Let's see, we've got another question for Dr. Lance regarding the UNCLE. How closely do UNCLE-derived TM values match those determined by DSC? Also, in the DLS data shown for case study 1, was the different traces in those plots due to different temperatures?
Okay, so I think for the DLS data question, I think that one was answered in a previous question where we mentioned that those are done at kind of room temperature. But addressing UNCLE and DSC, so that one's pretty interesting because UNCLE data correlates pretty well with DSC, but they're actually measuring slightly different things, where DSC will measure a protein physically moving and shifting in response to heating. UNCLE's measurement by fluorescence is optically looking at when different fluorescent domains of your protein are changing. So you can expect a little bit of a difference between those two behaviors. You could have movement that is not impacting any kind of fluorescent amino acid, and likewise individual fluorescent amino acids that move and shift, like say if you have an individual tryptophan residue that gets buried, that might not register as a very large event by DSC as well.
Okay, thank you so much, Dr. Lance. Let's see, we've got another question here. The first case study compares the characterization of 2 enzymes. Why do you only use 2? Why not 3 or more?
So I can take that one. The short answer is only 2 were available, and we were evaluating them post-purchase. So we observed different behavior in an assay and wanted to dig in a little bit to see why they were so different, even though they were nominally the same enzyme.
Okay, thank you so much for that answer. Let's see, it looks like we may have another question. How does B2S decide if advanced characterization is needed for a custom reagent?
So I can go ahead and take that one too. Essentially, we evaluated on a case-by-case basis. We try to fit our characterization plans as well to the client needs as well as what makes sound scientific sense. So for questions that are rooted in what a protein's overall structure might be, and whether it's folded, unfolded, whatnot, UNCLE is a good choice for that. But we also offer a number of other services that we use for different cases.
Perfect. Thank you so much. Let's see. Let me check. Looks like we have another question here. What would you say is an experimental limitation in UNCLE? Any constraints?
Okay. So one thing that comes up with UNCLE is if you're using these 3 detection methods, fluorescence, static light scattering, and DLS, the one that's pretty common and missing from that list is actually UV-Vis for concentration measurements. So if you have kind of a sample that maybe is coming off of a chromatography column, or there might be another reason why you aren't certain of its concentration, then that's something that you would typically check with UV-Vis. And so that's what we have other instruments like the Lunatic, where they take 2 microliters of sample and get concentration, and then you can take a further 9 and run that in UNCLE and get all these answers on stability and aggregation. So it's kind of just another technique that people really commonly use side by side with UNCLE. That's also helpful to know when you're kind of normalizing all these different experiments and samples together.
Perfect. Thank you for that answer. I have a few more questions. Let's see if this is worded. Does PEG harm the sample too, or can PEG harm the sample?
So it is totally depend on where we make the conjugation. So here we took a native enzyme and conjugated to a protein, to a PEG. So it's depending on the amino acid residue that's used for the conjugation is important for the stability. So for example, if there's a cysteine is involved and for the stability or the conformation of folding, and if you reduce the cysteine and conjugate it to a PEG, then that could potentially unfold your protein and reduce your stability. So for example, if you use a lysine that is on the surface and it is not essential for the stability, and if you use that to conjugate to a PEG, then you might not see such thermal stability issues was conjugated versus unconjugated molecule. So it is totally dependent on what is the residues that if you use to conjugate the molecules.
Thank you so much. It looks like we're out of time for questions. So thank you, Dr. Bowsher, Adam, Dr. Lance, and Dr. Abdeen. Do you have any final comments for our audience?
I would just like to say thank you, everyone, for attending. Lots of great questions.
Likewise, Adam, I see a number of friends online, and I would just want to say hello and thank you for attending.
Absolutely. Thank you very much for an engaging webinar.
Well, thank you again, Dr. Bowsher, Adam, Dr. Lance, and Dr. Abdeen for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Unchained Labs, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Please join us for Unchained Labs' next webinar broadcasting on February 11th at 8 a.m. Pacific, 11 a.m. Eastern. Keep it small, AAV and LNP quant and sizing from just microliters. We hope to see you there. Until next time, goodbye.